Chapter 7, Part 3, The Cell, Mitochondria and Chloroplasts. Mitochondria and chloroplasts are the organelles that convert energy to forms the cells can use for work. Mitochondria and chloroplasts are also some of the heaviest organelles, the densest organelles, found in eukaryotic cells. Remember that bacteria, or prokaryotes, do not have mitochondria and chloroplasts, as these are membrane-bound organelles. Mitochondria break down glucose to make ATP, a form of energy that the cells can use directly. Chloroplasts take sunlight to make their sugars, their glucose, basically using the energy from the sun and converting it into chemical potential energy found in the bonds between carbon and a glucose molecule. Both mitochondria and chloroplasts will make ATP energy. The end result of mitochondria is just ATP, whereas in chloroplasts they will make ATP in order to provide energy to make their carbohydrates, or stored energy in the form of glucose, represented by the hexagon on this slide. Mitochondria and chloroplasts have many similarities, and you should be familiar with these similarities. They both transform energy. They both generate ATP, even though the chloroplast will not use the ATP as an end result of photosynthesis, but as an intermediary step in order to make its glucose. In mitochondria, the ATP is the main reason why that organelle exists within us. They both have a double membrane, two membranes, an inner and outer membrane. The thylakoid stacks, shown with the cursor, are actually a third membrane found in chloroplasts. They're both semi-autonomous organelles, which means that they both act somewhat separate from the rest of the cell. They can move on their own, change shape, and divide independent of the rest of the cell. It's almost like a little cell living inside of a bigger cell that we're going to talk about later. Also, both mitochondria and chloroplast, mitochondria listed here, chloroplast picture over here, have their own internal 70S ribosomes, their own circle-shaped DNA, and make some of their own enzymes. Now, they're not completely autonomous of the rest of the cell. They do need some things produced by the cell in order to survive. However, they do have some similarities with free-living bacteria, which results in us thinking that perhaps they were once free-living bacteria a very long time ago. Let's start with the mitochondria. The function of the mitochondria is something called cell respiration, or cellular respiration. And the purpose of cell respiration is to generate ATP. Remember that the energy is stored in the unstable bond of the third phosphate group in ATP. The mitochondria will make ATP from the breakdown of sugars, fats, and other fuels like proteins. It doesn't have to use primarily sugar. However, that is the main source of energy that is broken down in order to make ATP. The other metabolic pathways to break down fats and proteins for ATP are much more complicated and are beyond the scope of this class. The mitochondria also only function in the presence of oxygen. They are going to break down the sugars in catabolism, or cutting it up into smaller pieces in order to harvest energy. This is an exergonic reaction. When the mitochondria make their energy in the presence of oxygen, this is called aerobic respiration. Perhaps you've heard of aerobics. When you do aerobics, you're literally using oxygen in order to break down the sugars in order to provide energy for the exercise in aerobics. The prefix "an" means without. Anaerobic respiration means without oxygen, which will be a pathway discussed later. The mitochondria has two membranes. It has a smooth outer membrane and a highly folded inner membrane, called the cristae. Like all things in biology, there is fluid, water, between the spaces, within the spaces, and surrounding the spaces. Water is everywhere in living things. The internal fluid-filled space, shown with the cursor right here, is called the mitochondrial matrix, not to be confused with the movie. This mitochondrial matrix is where the DNA, the ribosomes, and the enzymes are located within the mitochondria. You should know there is an example of the relationship of structure to function within a mitochondria. The second membrane, the highly folded inner membrane, increases surface area for membrane-bound enzymes that are going to make ATP. With more folds, you have more places for the proteins of the electron transport chain to make ATP to be located, which means that you can make more ATP. Here we have a transmission electron microscope photo of the mitochondria. As you notice, you can see all the inner folds called cristae in that second membrane that increase surface area for more chemical reactions to occur. So what's the significance of that inner membrane that's highly folded? Well, Chapter 9 is going to go into a lot of details, and this is a little preview of what Chapter 9 is all about. It involves a lot of coupled reactions and a lot of pumping of hydrogens into an inner membrane space. However, the carrier proteins located on the inner folds of this membrane are going to be absolutely critical to making lots and lots of ATP energy. Basically, what's going to happen is hydrogen ions, represented by the H pluses, are going to be pumped into this space in between the inner membrane and the outer membrane, located as dark orange. These highly concentrated hydrogen ions are going to flow from high to low concentration back into the matrix, or inner part of the mitochondria. And as these hydrogen ions flow through an enzyme called ATP synthase, this is going to drive the adding of a phosphate group to ADP to make ATP. This is where most of our energy from the mitochondria comes from. And if this is a little bit confusing, don't worry. We're going to go into more details in Chapter 9. Dividing mitochondria. Mitochondria divide by making a copy of their circle-shaped DNA, and then they divide the rest of their mitochondria. The only thing that acts kind of like that is bacteria. Bacteria do the same thing. They divide their circle-shaped DNA, and then the rest of the cell divides. Mitochondria divide just like bacteria. Using what we understand about nature, we can start getting clues as to where maybe this mitochondria came from. And it tells us that possibly these mitochondria were once free-living bacteria a very long time ago. Mitochondria. Almost all eukaryotic cells have mitochondria. There may be one very large mitochondrion, or hundreds if not thousands of individual mitochondria. The number of mitochondria is correlated with the aerobic metabolic activity. If you remember, aerobic means with oxygen. More activity means you need more ATP energy. More energy needed means that you need more mitochondria. I would like to point out at this point, need is kind of a misleading term. When I say a muscle cell needs more mitochondria, I'm really saying that nature would select for an organism with more mitochondria in a muscle cell because it provides more energy and allows the muscle cell to be more active, which would give a survival advantage to any animal possessing more mitochondria per muscle cell. The other type of cell with lots of mitochondria, in addition to muscle cells, are nerve cells. Nerve cells, which is basically what your brain's made out of, are very metabolically active. They require a lot of energy. It's estimated that when you're thinking hard thoughts, like, say, doing some math problems, you are burning off as much calories as walking slowly. I guess if you wanted to lose more weight during exercise, you would run and do math problems in order to burn off even more energy. Turn to your partner. Describe the structure and function of the mitochondria in eukaryotic cells. And what kind of cells have lots of mitochondria and why? Notice the mitochondria within the animal cell. If you saw a cell with lots of mitochondria, you would know it needs a lot of energy. We also went through the nucleus and the really tiny ribosomes. Notice that plant cells have mitochondria as well. People forget this. The mitochondria breaks down sugars for energy. The chloroplast, located in green, make the sugars to be broken down by the mitochondria later. Chloroplasts. Chloroplasts are plant organelles. They belong to a class of plant structures called plastids. If you remember, The fancy name for starch was amylose. So you can probably guess that amyloplasts are vacuoles that store starch. And you see a lot of these when you look at a potato cell, because mainly a potato is starch. Chromo means color, and a chromoplast stores pigments in fruits and flowers that give them their colors. Then we have chloroplasts. Chloroplasts store chlorophyll and function in photosynthesis. In leaves, they provide the green color. If you see a green stem, you know there's some chloroplasts in the green stem. You would not expect to find many chloroplasts in the roots of a plant because there's no light that can get there. Chloroplast. The structure is two membranes. It has an outer membrane and an inner membrane. However, unlike the mitochondria, the inner membrane is not highly folded, and nothing of importance, of significance, happens between the two membranes of a chloroplast, unlike the two membranes of a mitochondria, where you're going to generate a hydrogen ion gradient that's going to be helpful to make an ATP. There's an internal fluid-filled space called a stroma. The stroma is located right here. There are DNA, ribosomes, and enzymes in the stroma. The equivalent of the stroma in a mitochondria would be the matrix. That was the fluid-filled space inside the inner membrane. Another structure that we find in chloroplasts is called the thylakoids. The thylakoids are these little tiny pancakes. I like to think of them as pancakes. Each one of these pancakes is called a thylakoid. A stack of pancakes, a stack of thylakoids, is called a grana. Now the thylakoids are very important. The thylakoids are going to do the first part of photosynthesis that we'll talk about with Chapter 10. Basically, the light capturing events inside the chloroplast happen in the thylakoids. The thylakoids will produce two things, ATP, as well as another high-energy compound called NADPH. The second part of photosynthesis happens in the fluid-filled stroma, where we actually assemble our sugars from carbon dioxide. All that will be the topic of Chapter 10. The membrane-bound enzymes of the thylakoid, this is one of those stacks of pancakes, are where a bunch of chemical reactions will occur to make our ATP energy and NADPH. This topic will be discussed in detail in Chapter 10. Now that we understand the structure of the chloroplast, now we're going to talk about the function. The function of the chloroplast is photosynthesis. Photo means light. Synthesis means to put together. Photosynthesis literally means to put together using light energy. The thing that we're putting together is primarily sugars, glucose, that are going to be used for a variety of purposes within the plant, including making their cell walls and providing energy. Photosynthesis also generates some ATP, most of which is going to be used for the making of sugars. We have a transformation of energy. We're transforming solar energy, light energy from the sun, into a chemical stored energy between the carbon atoms of a glucose molecule. We're converting light energy into chemical potential energy within the sugar. We're going to be making our sugars, which are going to have the stored energy from the sun, from carbon dioxide gas and water. The chloroplast is also semi-autonomous. It moves, changes shape, and divides, semi-autonomous of the rest of the cell. It also reproduces by pinching in two, very similar to how bacteria divide, which is binary fission. Why are chloroplasts green? There are light-capturing pigments called chlorophyll within the chloroplast that will capture light energy that's not green light. The green light bounces off, and that's what your eyes see as green. It's the other colors that are not green that are actually being captured by the chloroplast and used for photosynthesis. So now we've discussed the mitochondria and chloroplasts, both found in plant cells, and just mitochondria found in animal cells. Mitochondria and chloroplasts are different from the other organelles. The organelles are not part of an endomembrane system, and we'll talk about what that means later. It's basically a transport system of delivering stuff out of the cell. The mitochondria and chloroplasts are semi-autonomous. They're semi-independent of the rest of the cell. The protein's primarily from the free ribosomes in the cytosol, but the mitochondria and chloroplasts can make a few of their own proteins from their own ribosomes. And they have their own circular chromosome, just like bacteria do. These are the clues that scientists have observed and have to try to make sense of. Remember, nature doesn't tell us how nature works. We have to figure it out. So who else has a circular chromosome not bound within a nucleus? The answer is bacteria. The endosymbiotic theory, developed in 1981 by Lynn Margulis, a female scientist from the University of Maryland. Mitochondria and chloroplasts were once free-living bacteria based on the evidence of some of the things that we see inside. We think that at one time, the ancestral mitochondria and chloroplast were engulfed by an ancestral eukaryote. An endosymbiont. Let's break down that word. Endo means inside. Symbiosis is a relationship of two organisms. Endosymbiont literally means the relationship of two organisms inside of each other. It's a cell that lives within another cell, a host cell, as a partnership with an evolutionary advantage to both. The mitochondria and chloroplast supplies energy, whereas the eukaryotic cell provides protection and raw materials needed to make that energy. Based on the observations of how mitochondria and chloroplasts work within eukaryotic cells, the endosymbiotic theory was developed to explain how mitochondria and chloroplasts came to be. Scientists believe at one time we had an ancestral prokaryote, probably about 3.5 billion years ago, based on fossil evidence and radiometric dating. We think that over time, perhaps millions of years, there was an infolding of the plasma membrane, the cell membrane, which resulted in some of the organelles with compartmentalized function that gave that eukaryotic cell a survival advantage. This ancestral eukaryotic cell, however, was not able to effectively use energy very well. We think that at one time, a free-living bacterial mitochondria was engulfed within a cell, the pre-eukaryotic cell. Now, normally when something gets engulfed within a cell, for example, our white blood cells engulfing a bacteria, there's hydrolytic enzymes to break it down, destroy it. And that probably would have happened with most cells that got ingested at the time. However, what we think happened is, at one point, an ancestral mitochondria did not get digested, and as a result, was able to survive within that eukaryotic cell. The mitochondria that was now free-living, still free-living, was able to divide independent of the rest of the cell and produce lots and lots of energy the eukaryotic cell can use. The eukaryotic cell benefited from this relationship because it was able to get more energy, ATP, than any other cell at the time. The mitochondria benefits because now it has protection within another cell type, and the other cell type, the eukaryotic cell, is able to provide energy, raw materials for energy, as well as protection from the external environment. It's this eukaryotic cell with the mitochondria living inside, once thought to be free-living bacteria, that gave rise to all heterotrophic eukaryotic cells, which includes kingdom fungi and kingdom Animalia. We think separate from the cell line that led to the animals and fungi was another event, a chloroplast that was also once a free-living bacteria, keeping in mind that blue-green algae are essentially what a chloroplast is that lives in modern times. However, what we think happened back in the day, probably about over 1 billion years ago, a chloroplast bacteria was engulfed by a cell that already had mitochondria, and now we have two things living inside of a cell: a chloroplast that can make sugars and a mitochondria that can break down the sugars to make ATP energy. This cell, actually a combination of 3 living things: the eukaryotic cell, the ancestral photosynthetic prokaryote that gave rise to the chloroplasts, and the mitochondria, all together keeping the cell alive and providing a pretty huge survival advantage. This is the cell type, mitochondria, chloroplast, and eukaryotic cell, that eventually led to all the photosynthetic eukaryotes, including all plants. From an evolutionary perspective, this is kind of a big deal. This is the first branching point between animals and plants. And this happened well before any plants or animals exist in their present form. One line developing into the types of animals and fungi we see on this planet today, way, way, way before there were plants and animals. And another cell type eventually leading to more complicated types of cells that can do photosynthesis that eventually led to the modern day plants. This happened well before 1 billion years ago. Interesting side note. Because mitochondria are passed down from females to their offspring, the sperm doesn't have mitochondria, it's all in the egg, we can trace maternal lines by analyzing mitochondrial DNA, the circle DNA within the mitochondria, back for as long as we have cell samples. So if you take a look at this family tree, this female here got her, well, all the children, males and female, got their mitochondria from their mother. So if we take a look back, when the mother, right here, and the father got married, it was the mother that gave the mitochondria to her children. And this female got her mitochondria from her mother, the grandparents of those children. As long as you have mitochondrial samples, or tissue samples in general, which have mitochondria inside of it, you can trace the maternal line back as far as you have cell samples. Turn to your partner and explain the endosymbiotic theory. Don't forget to add what was engulfed first, the bacterial mitochondria or the bacterial chloroplast. Also, explain what cells arose from these ancestral eukaryotic cells. Plants, animals, fungi, where did they come from? You should tell the person next to you. Ancestral prokaryote developed a nucleus, became the ancestral eukaryote. Basically a protective area for the DNA to reside in. Step 2. Ancestral eukaryote engulfed a prokaryote, bacteria that did cell respiration. This became the ancestor to all fungi and animal cells. And lastly, number 3. Ancestral eukaryote with mitochondria engulfed a prokaryote that does photosynthesis. This was the ancestor to all plant cells. Now, turn to your partner. What evidence supports the endosymbiotic theory? Without evidence, it's not science. The evidence that supports the endosymbiotic theory, number 1, mitochondria and chloroplasts divide independently or semi-independently of the cell. Number 2, the mitochondria and chloroplasts have their own circular bacterial-like DNA. And 3, mitochondria and chloroplasts have some of their own ribosomes and proteins independent of the cell. Turn to your partner. Compare mitochondria and chloroplasts. Compare means what are the similarities. You should tell them. Both produce energy. Both are semi-autonomous. Both make ATP energy, though chloroplasts do it to make sugars, and mitochondria make a lot more. And they're both found in eukaryotic cells, not in bacterial cells that are prokaryotes. Contrast, or what are the differences between mitochondria and chloroplasts? Mitochondria found in all eukaryotic cells, whereas chloroplasts like plants, and some protists like algae. Mitochondria break down glucose to make ATP, whereas chloroplasts build sugars up by using light energy. It's a different type of transformation of energy. Any questions?